Well, hi everybody. This is our virus lecture. Viral genetic systems is what we'll be talking about today. We're still in Chapter 8. We're going to finish off the second half of Chapter 8 today, which is pages 219 to 230. We're going to start off talking about viruses that infect bacterial cells. These are, for historical reasons, referred to as phages or bacteriophages. So, as is often the case, we'll set the stage for how complicated things like viruses work in the simplest way possible, how they work in bacteria. We'll go back to the discussion of transduction that we began in the last lecture. We'll define two more specific types of transduction, generalized transduction and specialized transduction. We'll describe how those two pathways work. We'll talk about how genes used to be mapped with these bacterial viruses, so you could use certain tricks and certain statistical measures to map genes that are on the viral genome using these bacteriophages. And we'll end the lecture with a discussion on RNA viruses, retroviruses. And the reason why is because these are the viruses that play such an important role in our own human health. HIV is an RNA virus. Influenza is an RNA virus. So we discuss those at the end of the lecture. So viruses infect every living thing out there. There's no cell type, there's no species on this planet that is immune from viral infections. And what's interesting is we can't even really classify viruses as living themselves. The best way to characterize or define a virus is to call it a genetic parasite. So think about what parasites do. Parasites feed off of or hijack their host systems in order to survive. So you can think of a tapeworm as the classic parasite. The tapeworm lives in the host's gut and absorbs nutrients from the host the virus feeds off of and thrives at the host's expense. A virus does the same thing, but a virus is not alive. Instead, a virus is really nothing more than a genetic particle or a genetic package. And the genes of the virus survive and thrive, if you want to think of it that way, at the expense of a host cell. So a virus is nothing more than genetic information. It can be DNA or RNA, some type of nucleic acid, surrounded by a protein shell. And the virus is then capable of either entering a living cell or injecting its genetic material into that cell. That genetic material then hijacks the cell's own enzymes and machinery, forcing that host cell to stop doing what it's doing for itself, but instead make many, many new viral genomes and many, many new virus proteins, protein shells. And then, with so many new viruses filling this host cell, the host cell is swelled to the bursting point. It lyses or breaks open. When it lyses, it releases all of those new viral particles that then go off to infect new cells and repeat the process again and again. So a virus is a replicating particle. It's protein and nucleic acid that is capable of replicating or copying itself, but it can't do so without a host cell. Indeed, a virus's entire reason for existence is just to propagate itself. It is a genetic parasite. I already said it on the first slide, but viruses that infect bacterial cells are called bacteriophages, or phages for short. And bacteriophages, perhaps actually more than any other thing studied in the life sciences, has played an enormously critical role in allowing us to understand and discover how genetics works, how inherited traits work, and indeed how molecular biology and molecular genetics works. The process I describe above here, the idea of a virus infecting a cell, hijacking the cell's machinery, making new viruses, and bursting that cell, that's referred to as the lytic cycle. The lytic cycle of the virus. And viruses that do this, have this lytic cycle, are called virulent because they cause symptoms. All these host cells bursting and dying causes symptoms, causes the host organism to become sick. So we call that a virulent virus. And I think this figure shows what I just said in a really nice, thorough way. We start off with our host cell. We have our host cell genome, and a phage, or a bacterial virus, has landed on the surface of that cell. Step 2 is the injection of the genetic material, whatever that genetic material is, into the host cell. Step 3 is the disintegration of the host genome, and this is what causes the host cell to stop doing what it was doing for itself. Step 4, the host cell machinery is hijacked, allowing new viral genomes to be made, as we see here in purple. Step 5, we make new viral proteins. Those cause or allow viral shells to be made, and the genetic material, the viral genomes, are loaded into those viral heads, those viral shells, creating new viral particles in the host cell. And finally, step 6, the host cell is so swollen with new viral particles that the cell membrane gives way, the cell bursts open, and the new viral particles are released. And each one of those new viral particles is going to land on a new host cell and repeat the process again. Now we have virulent viruses, but we also have temperate viruses. Temperate viruses aren't quite so mean. They aren't quite so destructive. And the reason why they aren't so destructive is because temperate viruses also have what is referred to as a lysogenic cycle. The lysogenic cycle begins the same way. We still have a viral particle landing on a host cell. We still have the injection of the viral genome into the host cell. But then, instead of destroying the host cell genome, the viral DNA integrates itself through crossing over events, recombination. We should be pretty comfortable with this by now. The viral genome in purple integrates itself into the host genome, very much like our plasmids integrated themselves into our bacterial host genomes, creating HFR strains. Same exact mechanism. The virus can stay integrated into the host genome for quite some time, in fact, well beyond this bacteria dividing through asexual reproduction. So every time the bacteria copies its genome, it is also, of course, going to copy the viral DNA, because the viral DNA is physically connected to the genomic DNA now. So this one virus divides into 2, and each of those new viruses has a viral genome integrated into it. These 2 cells can divide into 4, copying the viral genomes again, etc. Now, sooner or later, and I said all that, sooner or later, typically when times get hard for the host cell is starving for nutrients, it's under some other type of environmental stress, these hard times, these stressors, serve as a trigger for that integrated viral genome to pop back out or excise itself, as we saw in the F' plasmids, excise itself from the bacterial genome, and then it enters the lytic cycle. So once excised, those viral genomes then cause the disintegration of the host genome, make many, many copies of the viral genome in that one cell, etc., etc. We continue on with the lytic cycle. So by using the lysogenic cycle, viruses have a mechanism by which they can actually hide and lie dormant within the host genome, being copied, still replicating, still being copied. Every time the host cell divides, the viral genome is dividing, but hiding because it isn't causing any symptoms, it isn't causing any cell destruction, it's causing no harm to the host cell. Any integrated lysogenic viral genome is referred to as a prophage, because it's not an active virus, it's almost like a virus in hibernation. So now, with that background on what viruses are and how they work, let's talk a little bit about how we were able to learn so much information through the study of these bacteriophages. Now, it stands to reason, and hopefully it's somewhat obvious to you at this point even, that viruses can't grow by themselves. They're not living things. Viruses have no ability to replicate themselves on their own. They can't grow. Instead, viruses have to be given host cells which they can infect and replicate within the host cell. Again, this is a parasite, so it can't survive on its own. It requires the hijacking of host cell machinery in order to carry out its life cycle. So in the lab, if you want to study and work with and grow bacteriophages, you need to grow them in bacterial cells. You need to intentionally infect bacterial cells with these phages in order for your viruses to replicate. So typically how this is done is on a petri dish, you plate bacterial cells in very high concentration. So high concentration, in fact, that the entire plate is covered with bacterial cells. I'm not sure how many of you have experience with petri dishes and bacterial media, but usually the media or the food that is in the petri dish for the bacteria is clear. And as you can see in this picture, it is not clear. We can't see through this petri dish. The reason why is because the entire surface of this petri dish is covered entirely with bacterial cells. We refer to this as a lawn of bacteria. So very much like the grass covers the entire front yard, the bacterial cells cover the entire petri dish. So there's very, very high concentration. Oftentimes when you're doing work with bacteria in the lab, you grow what are called colonies, small, round, individual collections of cells that are about the size of a pencil point. And they are scattered around the plate, leaving the rest of the plate, where there are no colonies, clear. But that's not what we have here. We have a lawn. Then you spray or add a small, low concentration of phage to that bacterial lawn. And some of those viruses, some of that phage, will infect and lyse a bacterial cell. Now remember the life cycle of the lytic cycle. Once this bacterial cell that was infected lyses, it's going to release many, many new viral particles that can then infect and lyse the bacteria in that area. When those bacteria lyse, you're going to release even more viruses that infect the next range or round of neighbors. And this continues on and on. So if we imagine a lawn of bacterial cells such as this, we spray some phage on, that phage infects a single bacterial cell and lyses it. That creates a microscopic pinpoint emptiness on the plate where there is no more bacterial cell. But we release hundreds of viral particles when that single cell lyses. So those new viruses are going to infect all of the bacterial cells around this original void from that first cell. And once those cells lyse, we clear that region of bacteria. But we've released that many more viruses which infect the next round, and then the next round, and then the next round. So we get these circles of cleared areas on these plates, on these bacterial lawns, that represent the dying and bursting of bacterial cells as the result of continuous phage infection. These are called plaques. So a plaque on a plate is a cleared region of cells due to some viral infection. And this is a real-world example of a plaque. You can see this plate started as a lawn of bacterial cells, just like the one I showed you earlier. But now we see all of these almost perfectly round spots where there are no longer bacterial cells present, and so we can see through the bacterial lawn to the plate below. These spots are cleared of bacterial cells because those bacterial cells have been lysed or burst open by an active viral infection. Pretty interesting stuff. So now with all of that in mind, I want to go back to the transduction material that we began talking about in the last Bacterial Genetic Systems lecture and give that a little bit more meat, give that a little bit more detail, because we now should have a pretty good understanding of bacteria and also now background on how viruses work. So let's bring the two together and discuss transduction a little bit more thoroughly. So in that last lecture, we briefly described transduction. We defined transduction as using viruses to transfer bacterial genes horizontally from one bacterial cell to another as a mechanism for bacterial genetic transfer. But let's explore transduction a little bit deeper. There are two types of transduction. Generalized transduction is the first that we'll discuss, and actually what I described in the last lecture is generalized transduction. This is when virtually any bacterial DNA can be transferred by a viral particle that has loaded bacterial DNA into its protein shell instead of its own viral genome. As we've already said, the lytic cycle involves a virus using bacteria to make new viral genomes and to make new viral protein shells, and then those two are packaged together, the genomes go in the shells, and those are your new viral particles. When those become too great in number, the host cell bursts. But as this is happening, and as I've already said, the bacterial cell is dying, and part of that dying process for the bacterial cell is that its genome is fragmenting. The genome, the bacterial genome, is breaking up into small pieces. Some phage, called transducing phage, that is phage that do transduction, make mistakes. And they accidentally package bacterial DNA, that fragmented bacterial DNA, into their protein shell instead of the viral DNA. So this is what we're talking about in pictures. We have our normal infection. We've seen it before. Here's the viral particle on the cell membrane. Here's the injection of the viral genome. We have the fragmenting of the host genome in red. We have the copying of the viral genome in purple. What should happen now is that all of the protein shells of the virus should be loaded with purple viral genomes. But every once in a while, purely by accident, instead what gets inserted into the viral protein shell is a fragment or piece of the host genome, here in red. So now this is bacterial DNA. This red DNA is bacterial DNA. in the viral protein shell. When this host cell bursts and releases all of these viral particles, when this red bacterial DNA carrying viral particle lands on another bacterium, it's going to do the only thing it knows how to do. It's going to do the thing it was programmed to do. It's going to inject that bacterial DNA from this cell over here into this cell over here. That bacterial cell DNA, of course, is non-infective. It's not viral DNA. It's not going to launch any infection. And that bacterial DNA is often integrated or recombined into this host cell. And so now we've had horizontal gene transfer for generalized transduction. So when this new virus, this misloaded virus carrying bacterial DNA, goes off and infects another cell, it injects that foreign bacterial DNA into its new host cell. We get an integration event. That DNA, that red DNA, is stably maintained. That is horizontal transfer of bacterial DNA from one individual to another. The bacteria that receives this foreign DNA, the bacterial DNA that has been the recipient of this transduction, is called a transductant because it was involved in a transduction. Lots of mistakes have to happen for this to occur. We have to have the misloading of the viral particle. We have to have that viral particle be competent to try to launch a new infection. We don't always get this crossing over event, this recombination, so that must occur for this DNA to be stable and maintained in the transduction. And so generalized transduction is a very rare event. Too many things have to go wrong for this to happen. Also, a viral shell is very, very small. Viral genomes are small, so viral shells are small. So the piece of bacterial DNA that can fit in that viral shell is exceedingly small. Only about 1% of a bacterial genome can fit into a virus particle. Finally, since the bacterial genome in that first infected cell was fragmented randomly, and that's what allowed a piece to be small enough to be loaded in a viral shell, if two genes are transduced together using generalized transduction, it stands to reason, in fact it has to be the case, that those two genes were next to or near one another on the bacterial genome when we had the initial infection. You can only get one piece of bacterial DNA into a viral shell. So if two genes are transduced in this way, those two genes must have been on the same piece of DNA. And since that piece of DNA had to be so small for it to ever fit in the viral particle in the first place, it means those two genes that were transduced together must be close together on the bacterial genome. And in fact, this reasoning, this rationale, was used in the early days of molecular biology as a strategy for mapping bacterial genes and trying to figure out which bacterial genes were next to which. But there is another type of transduction called specialized transduction. Still involves viruses, but in a bit of a different way. In this form of transduction, the virus infects the initial cell, but then instead of going lytic, the virus enters the lysogenic cycle. And so the viral DNA is integrated into the host genome. When the virus pops out and excises itself out of the host bacterial genome in order to start to begin the lytic cycle, a mistake is made. And accidentally, some parts of the host genome DNA are taken or excised with it. Very much like that F' plasmid that we talked about initially, taking some of the bacterial genome with it. Because of how this mechanism works, the virus only takes bacterial DNA that was closest to its borders at the integration site. That's why it's called specialized, because not any bacterial DNA can be transferred in this way. Only bacterial DNA next to and adjacent to the integration point of the virus can be taken. Only specific DNA is transduced here. Once that excision is complete, even when it's in error and some bacterial DNA is taken as well, the viral DNA, along with that bacterial DNA from the border, will be loaded into a viral protein shell. The host cells will eventually burst, the viruses will go off and infect a new bacterial cell, and if and when that new infection gets stably integrated into the new bacterial cell's genome, we have completed the transfer of bacterial DNA horizontally. That's why we refer to this as transduction. Again, I can't wait to get to the pictures with these things. I'm a visual learner, so pictures for me drive things home a whole lot more than words spoken or text on a slide can. So this is what we mean. We're skipping all the early steps. I have some confidence that you're comfortable with that by now. So we're already in the lysogenic cycle. We've had the viral infection. We've had the integration. We are now in this prophage state where the viral genome has integrated into the host genome. So we've integrated. And now times get hard. The cell begins to starve. And so the viral genome wants to excise. It wants to pop out. It wants to go into the lytic cycle. So as the viral DNA begins to excise itself from the host genome, it accidentally takes some of the host DNA with it, and in turn leaves behind some of its own DNA. It can only fit so much into that viral head. But keep in mind, the host DNA that was taken had to have come from the borders of the insertion site, because that's the way the viral particle, the viral genome excises. In other words, it's specialized transduction because you can never, ever hope to get any of this bacterial DNA loaded into a viral particle because this bacterial DNA is so far from the viral insertion site. Only border sequences on either side of the viral insertion site have any hope of being transduced in this way. Once excision is complete, however, that viral genome is loaded up into viral protein shells. Those viral protein shells get to such a sufficient number that they burst the host cell. Those viral particles now are released and go on and infect new host cells. And when they do so, especially if they integrate, they transfer the bacterial DNA from the border region of the initial cell into the genome of the new host cell, and that is your transduction. That is your horizontal gene transfer. So let's come back to this idea of gene mapping and phage. By now you probably appreciate the fact that mapping genes is a critically important part of genetics, and so was the case well before the ability to sequence DNA. Knowing where genes were on a physical molecule and where they were positioned relative to one another, who was next to whom, we could learn quite a bit about inheritance patterns, and we could do very high-level predictions about inheritance patterns in genetic matings. So, mapping genes using phage, kind of not surprisingly, requires a lot of the same principles and a lot of the same rationale as mapping genes in any other organism, as mapping genes using recombination frequencies. In essence, gene mapping in phage is due to recombination events. And so all that we learned about recombination frequencies and crossing over events there will apply to everything we're going to talk about here. In essence, we're not going to be learning anything new in these slides. We're just going to be reapplying that information in a new and novel way. So let's consider this scenario. We have two viruses. Each is mutated in such a way that it can no longer cause infection. And so we have two viruses that are not virulent because of their mutations. And we use these two viruses to co-infect a bacterial cell. In other words, we infect a bacterial cell with both of these mutant viruses, neither of which is capable of launching an infection, simultaneously. When recombination happens, a new virus will be made that contains no mutations. So we'll get one viral genome that has no mutations, and we'll get another viral genome that has both mutations together on the same physical DNA molecule. Now, the initial mutated viruses were avirulent because of their mutations. Certainly, the new viral genome with both mutations together is also avirulent. But the other new viral genome we made as a result of this, with no mutations on it, is virulent and can launch an infection. By doing these types of studies, we can easily map the distance between viral genes. I'll give you an example. The classic experiments that were done this way used a specific type of bacteriophage called T2. It's just the name of that virus. One T2 strain could infect and lyse cells of the B variety in bacteria, but couldn't infect or lyse bacterial cells called B2. Also, when infections occurred, the plaques made by these mutant T2 viruses were distinct. They had sharp borders, not fuzzy borders, but sharp borders of their plaques. Another T2 strain, which was not mutated, could infect both B and B2 bacteria, but it made normal wild-type plaques with these fuzzy borders. So really what this boils down to, get away from all the details. You're probably feeling overwhelmed at this point. A lot of terminology, a lot of letters, not making such sense. We're really just dealing with two traits. One trait is the ability to infect B and B2 cells, or B cells only. The ability to affect B and B2 cells is different and distinct from the ability to infect B cells only. You could think of this as a phenotype for that gene. Our other trait is the appearance of the plaques these viruses give. We can have these sharp-bordered plaques or these fuzzy-bordered plaques. That's our phenotype. So we're looking at two traits with two measurable phenotypes each, and we can use that almost as a dihybrid study where we look for recombination between these two viral genes, these two genes on T2. So here's how the experiment was done. Bacterial cells were infected with both of these two T2 strains simultaneously. And the experimenters saw lots of viruses that can only infect B cells, which had distinct borders. Only infect B cells with distinct borders. Well, we started with that virus, and we see it come back. And the experimenters saw lots and lots of viruses that could infect B and B2 bacteria with fuzzy borders. Well, can infect both B and B2 bacteria with fuzzy borders. We saw that too. We added that virus. Of course, we're going to see lots of plaques that way, because we added that virus. But the experimenters also saw viruses that could only infect B cells and gave fuzzy bordered plaques. We didn't add any viral particles like that. We didn't add that phage to our cells. And the experimenters saw some, not many, but some viruses that could infect both B and B2 bacteria that gave distinct sharp-bordered plaques. And we didn't add any of those viruses either. So the question became, what happened? We co-infected with two viruses. Those viruses had two traits each. We saw lots and lots of plaques from one of the viruses we used. Of course, those viruses infected bacteria. We saw lots and lots of plaques from the other virus we used. Of course, we used that virus to infect bacteria. But we also saw viruses that gave plaques that appeared to swap these traits among each other, almost like they traded them. Here, you take my fuzzy border gene, and I'll have your distinct gene. And we didn't add those viruses. So what happened was recombination, just as we saw it happen in our earlier lectures. Recombination occurred a very small percentage of the time when both T2 viruses were co-infecting a single bacterial cell, and that's key. Both viruses had to be in the same cell at the same time. We got recombination, and we swapped that genetic information. So if you think of this as B only and distinct borders, and you think of this gene as B or B2 with fuzzy borders, we started with a genome that was B and B2 infection with fuzzy borders. We added that, we started with it, and we got lots of it back. We added a viral genome that was B only with distinct borders. We added that virus infected, and we got a lot back. But we also got recombination. And when we got recombination, we created new viral genomes that were the ability to infect B and B2 cells with distinct borders and the ability to infect B cells only with fuzzy borders. And that was the result of recombination between those two molecules. Now, by counting up the number of recombination events, same as we did before, by counting up the number of recombination events, how do you count up the number of recombination events in this experiment? Well, you count up the number of B-only fuzzy-bordered plaques and B2 distinct bordered plaques, because those plaques come from recombinant viruses. So in fact, by counting up the number of recombinant plaques and then dividing that by the number of total plaques, scientists could calculate a recombination frequency between these two genes. And, as we know by now, this recombination frequency can be used as a physical map, expressing the distance between those two genes in centimorgans or map units. So that's great. That gives you the basics, the background of how we can use recombination techniques to map genes in Phage. But Phage offered something that no other model system had offered up until this point. Huge, huge numbers of progeny. Huge, huge numbers of events. With that large number of events, because so many viruses could be made, scientists were able to pick out the rarest of the most rare occurrences, because they simply had so many samples to look at. In the 1950s, molecular biology did not exist at all. There were no experimental techniques for studying DNA. In fact, the structure of DNA had just recently been determined, and DNA was simply known to be the heritable material of life. That was it. But technology had not caught up with all that DNA had to offer. So how DNA worked still remained a mystery. During this time, the largely held consensus was that a gene, still an abstract idea representing an inheritable trait, was that a gene was the smallest fundamental functional unit of DNA. In other words, you could chop DNA up into lots of little pieces, but you couldn't, they thought, chop DNA into pieces smaller than a single gene. They thought a gene, a single gene, was the smallest piece of DNA that you can get. We know now, of course, that that's not true. Genes are nothing more than sequences of nucleotides, sequences of A's, T's, G's, and C's. And so it is the single nucleotide, much smaller than a gene. It is the single nucleotide that is the smallest functional fundamental unit of DNA. But they didn't know that at the time. And the thing that allowed scientists to realize that you could go smaller than a gene, that genes could be chopped up themselves, was research using Phage. Research looking at recombination in Phage. Using techniques to measure intragenic mapping. What do you think that means, intragenic mapping? Remember, interstate highways cross state lines. Intrastate highways, if there was such a word, would remain in a single state. When you're doing intragenic mapping, you're mapping genetic distances within a single gene. So the ways these experiments were done, two mutations within a single gene were genetically mapped using recombination techniques. Pioneering work. We introduced the idea of complementation in an earlier lecture. I'll review it here, though, if you don't remember how it worked. The way that scientists know if two different mutations in two different genes are occurring, and how they distinguish that from two different mutations in one single gene, is through complementation analysis. If the mutations were in two different genes, co-infection, if we're thinking in terms of phage, co-infection with the two mutants would result in plaques on cells every single time because of complementation. Here is one viral genome with two genes on it, A and B. Here's another viral genome with the same two genes on it. And keep something in mind here. Genes, again, are nothing more than the instructions for building proteins. We've said that a number of times already, but remember that now. And it's the protein that is the machine that carries out whatever essential function is needed. It's the protein that's important. So you can imagine a scenario for whatever reason. You don't have a gene for a specific reason or a specific purpose, but you were given the protein. Artificially, experimentally, the protein was inserted into the cell. If that's the case, if you have the protein, you can carry out the function. The protein is what matters. The gene simply allows you to make that protein. So now I'd like you to imagine that one of these viral genomes has a mutation in gene A, and the other viral genome has a mutation in gene B. The proteins made from gene A and gene B are both required to launch this viral infection. So the first virus up here can't infect a cell on its own because it can't make the A protein. The second virus below can't infect a cell on its own because it can't make the B protein. However, if we co-infect a single cell with both mutant viruses, we do see infection. Why? Because we are making the protein from gene A off of this viral genome, and we're making protein off of gene B off of this viral genome. Protein A and protein B are both present in the host cell now, and so the viral infection continues. That is complementation. Now, I'd like you to contrast that with the mutations both being in the same gene. When the mutations are both in the same gene, we start off with the same story. The first virus on top can't launch an infection because it has a mutation in gene A. It can't make protein A. The virus on the bottom can't launch an infection because it has a mutation in gene A. It can't make protein A. We can co-infect with these two viruses and get both viruses simultaneously in a single host cell, but we still don't see infection because still protein A is not made in that host cell. And we need protein A for an infection. But if we get a recombination event, if crossing over occurs between the mutations, we're going to have a genetic swap. We're going to take everything from before the insertion, the recombination event, and swap it with everything after. And what that's going to do is bring this entire bottom piece up to the top genome and this entire bottom piece down to the bottom genome, resulting in a switch or swap of the mutation as well. So now what do we have? Now we have a viral genome with two mutations in gene A, so it still can't make protein A. That viral genome cannot launch an infection on its own. But we also have a viral genome with no mutations now, fully capable of making protein A and protein B, and so now we see infection. But because these two mutations were in the same gene, complementation is not what happened. It's recombination that allowed the infection to occur. This is very, very rare because recombination tends to be rare events, and it happens much less than the 100% of the time we would expect to see if complementation was our cause. So recombination in general is rare. Recombination between genes that are close together is even more rare. Recombination between specific nucleotides in a single gene, exceedingly rare. And again, only phage, only these types of genetic studies give large enough numbers of progeny where we can pick out these extremely rare events. For these experiments, another bacteriophage called T4 was used. Wild-type T4 with no mutations gives small plaques with rough edges on the plaque when infecting E. coli. So you can see examples of that in the center here. Rough edges, not distinct, and the plaques tend to be smallish. Some specific classes of T4 mutants, mutations in some of the genes called R mutants, because they have mutations in their R genes, give plaques with a different morphology. They give plaques that tend to be a bit larger, but more importantly, the edges of those mutant plaques from the mutant phage are sharp and distinct. The experiments that were done focused on a subgroup of those mutants called R2 mutants, because that's the gene where the mutations occur. R2 mutants produce large, sharp-edged plaques on B strain E. coli and cannot at all infect K strain E. coli. That's the mutants. Of course, wild-type T4 can infect both B and K strain E. coli cells and give that wild-type shape to their plaques, small and fuzzy-edged. The experiment that Benzer did to study and map intragenic distances was a simple one. He co-infected E. coli cells with different R2 mutants, and he looked for a reversion to the wild-type T4 pattern of the ability to infect K cells with small and fuzzy borders. The only way that two T4R2 mutants, remember R2 is the gene, so the only way that two different mutants in the same gene could give rise to a plaque with a wild-type shape and give rise to an infection on K cells is if a recombination event occurred between them when they were mixed together and co-infecting a cell. So here was the Benzer experiment. Benzer, again, started off with the initial prediction. Mutant number 2 could infect B cells, but could not infect K cells. So after injection into K cells, no plaques were found. Mutant number 1, mutation still in the R2 gene, but in a different part of the R2 gene. Mutant number 1 could infect B cells, but could not create plaques on K cells because of that mutation. Since both of these mutants could infect B cells, Benzer co-infected B cells with both bacteriophages simultaneously. See, Benzer had to get both molecules in the same place at the same time to give recombination a chance to occur. And he couldn't use K cells because neither mutant was capable of infecting K cells. So Benzer infected B cells with co-infections, getting both of these mutants into the B cells simultaneously. He let those B cells lyse, he collected the phage that resulted, and he used those phage to try and infect K cells. And lo and behold, some very small percentage of the time, he could get K cells infected. What allowed the K cells to be infected here, but not here or here? A recombination event, which put both of the mutants together on one viral genome, but left completely non-mutated DNA for the R2 gene on the other viral genome. And these non-mutated viral genomes is what was allowing the infection on K cells. So all Benzer did at that point was count up the number of plaques on his K cells plate. He then multiplied it by 2. Why do you think he did that? Why do you think he multiplied by 2? Why would he have to double his number? Remember, we would like to count up the total number of recombinants after this. The total number of recombinants and divide it by the total number of phages we were looking at. When Benzer counts up his plaques on the K cell plate, 1, 2, 3, what is he counting? He's counting 3 viral genomes that look like this, because only these viral genomes can give those 3 plaques. But what we need to count is the total number of recombinant viral genomes. The total number of recombinant viral genomes. We need to include these guys in our total of recombinant, but we don't see them. They never give plaques on a K cell plate. So how can we count something we can't see? Well, what we can do is realize that every time we make this viral genome, we make one of these 2. So if we see 3 plaques, we know definitively we made 3 of these viruses, but we also know hiding out in the background are 3 of these as well. So we double our 3 to 6, and we count our total number of recombinants. He then divided that number by the total number of plaques on B cells, which represented all the viruses that were harvested at all the total number of progeny. And by doing this, he was able to calculate a recombination frequency within a single gene. This work provided the first molecular view of a gene. It showed that recombination could occur between portions of a single gene, thus proving that it was physically impossible for a gene to be the smallest functional unit of DNA. Seymour Benzer unlocked a bridge between a genetic world and a DNA world, and really paved the way for modern molecular biology and for thinking about genetic molecules, genetic concepts in a molecular and DNA-based way. Just amazing. So now I want to start the winding down of the lecture by introducing our last topic. I want to talk a little bit about RNA viruses because, as I said in the very first slide, they play such an important role and devastating role in overall human health. So viral genomes for viruses come in all shapes, sizes, and types. We have double-stranded DNA viruses, single-stranded DNA viruses, single-stranded RNA viruses, and even, rarely, but they exist, double-stranded RNA viruses. RNA viruses themselves as a subfamily include some of the most important viruses from a human health perspective, and most RNA viruses fall into a class called retroviruses. Retroviruses are the classic virus. They're very, very durable. They tend to be made up of RNA genomes, by definition, covered with a protein coat, a protein shell, then covered with membrane, cell membrane, that had been stolen from previously infected cells. The flu, influenza, is an example of a retrovirus. The common cold is a retrovirus. Polio is a retrovirus. And, of course, HIV, human immunodeficiency virus, is a retrovirus. RNA viruses have a kind of lysogenic cycle. It's a little bit unique because there's that RNA to contend with. Most RNA viruses and all retroviruses, when they infect a host cell, the RNA genome is first reverse transcribed into DNA. If you remember from our extreme early lecture, our introduction lecture to the whole course, we defined transcription as the process of copying a DNA gene into an RNA molecule. Well, reverse transcription goes the other way and is capable of copying RNA back into DNA. This DNA copy of the initial viral genome, now in DNA form, is capable of integrating into the host genome, resulting in a prophage, kind of like a provirus, kind of like a prophage, except we don't use the word phage because now we're talking about viruses that infect us, resulting in a provirus that is dormant and hiding, integrated into the host genome. When a signal is received by the viral genome that it's time to move on, again, stress on the host cell, stress from the environment, many, many triggers for this, that integrated viral DNA-based genome is transcribed through the standard process of transcription using the host cell's enzymes. That's what always pisses me off. It's your RNA polymerase, your enzyme, which is copying this virus's genome for it. Your own cellular machinery is being fooled and tricked into doing something that is going to cause you harm by this genetic parasite who specializes in such trickery. That transcription results, as all transcription does, in making an RNA copy of the DNA that was transcribed. Since the DNA that was transcribed here was the DNA version of the RNA of the viral genome, transcription makes lots and lots of RNA copies of that viral genome. Well, since this was a retrovirus, this is an RNA virus, those transcribed RNA copies of the viral genome are new viral genomes. These viral genomes are then packaged into protein shells. Where did those protein shells come from? They came from those same viral RNAs. Just like our own RNAs can be translated into protein, according to the central dogma, transcription gives you an RNA copy of a DNA gene. Translation gives you a protein from that RNA. Our own cellular machinery makes the viral protein as well. Those viral proteins assemble into a viral cap, viral shell. The RNA viral genomes are assembled into that shell, and then the virus buds off of it. Doesn't lyse the host cell. Our viruses evolved to be much smarter than that. No point killing the host cell when you can manipulate it further and get more out of it. And so the viral particle buds off of the host cell membrane, stealing some of that host cell membrane with it. In fact, the theft of that host membrane makes it much, much easier for that virus to go on and infect another one of our cells, because that host membrane covering that virus fools other cells into thinking that the virus belongs, that it's one of us, one of our own cells. Okay. So I'll just review the life cycle very quickly. The virus lands. Because it's covered in host cell membrane, it's not recognized as foreign. The virus merges with or inserts itself into the host cell. The RNA viral genome is liberated and released. It is reverse transcribed into a DNA copy. That DNA is then integrated into the host genome, and it lies there indefinitely in a dormant state. Once triggered, that viral DNA genome is transcribed by host enzymes into an RNA copy. That viral RNA serves two purposes. It is, one, a viral genome, and two, the instructions for building viral proteins. Translation of that viral RNA results in the manufacture of viral proteins. Those proteins assemble into a new viral shell. The RNA viral genome gets inserted into that shell, and then that protein shell buds off of the host cell, stealing some membrane with it to repeat the cycle somewhere else. All known retroviruses on Earth, all of them that have been studied, have three main genes in common. The GAG gene, this is the gene containing the instructions for building the viral proteins that make up the particle shell. The PAL gene is the gene for making the reverse transcriptase enzyme, which is responsible for that initial reverse transcription, converting the RNA genome into DNA. Also, reverse transcriptase aids in the integration of the viral DNA genome into the host genome. And then the envelope gene, or ENV, which is used for making sugar-coated proteins that appear on the virus's surface, poking through the stolen cell membrane, which is another layer of foolery that the virus uses to convince host cells that the virus belongs and is one of us and is self. So HIV and AIDS. Well, HIV, or again, human immunodeficiency virus, causes the disease-acquired immune deficiency syndrome, or AIDS. So we always say HIV is the virus that causes AIDS. An individual can be infected with HIV, yet not have AIDS because they're completely asymptomatic. We'll talk about why in class. There are two different viruses that cause AIDS. They're classified as HIV-1 and HIV-2. Together, these two viruses have infected more than 65 million people worldwide, and about a third of them dying as a result. You know, it's a horrible situation and a widespread issue and disease, but it is important to say that that lethality rate is actually not so bad. There are some viruses with lethality rates a whole lot higher than 33%. HIV-1 is the main HIV, if you want to think about it that way. It's found globally. It's responsible for the majority of cases that we see worldwide. HIV-2 is still largely confined to the Western African continent. It's believed, more and more evidence is supporting the fact that HIV is likely to be a mutated version of simian immunodeficiency virus. This is the virus that infects chimps and other bonobo chimps, other primates closely related to us on the evolution tree. Interestingly, these chimps are completely asymptomatic. AIDS-like diseases, or I should say SIV viruses in chimps, has evolved to a point where it can exploit the chimp as a host, see to its own replication, but do so with as little harm to the host cell and the host organism as possible. So chimps contract this disease, they spread this disease, but since they're asymptomatic, it really does them no harm. It's further likely that two different SIV viruses simultaneously co-infected a few chimps, and because there was co-infection going on, recombination had a chance to occur, and it did occur, creating a new novel virus that was ideally suited for infecting human beings. So we have two different types of SIV, one more suited to mangabees and the other spotted to a spot-nosed monkey. Both of these viruses was capable of infecting chimps, and it's believed that somewhere along the line of evolution, both of these viruses co-infected single chimp cells in some highly infected individuals. Recombination occurred between these two different SIV viruses, giving rise to a new and novel SIV virus that hadn't existed before. This new novel SIV virus was also spread among the chimpanzee population, and somewhere along the line, this SIV virus made the leap from infecting a chimp to infecting a human being who had come in contact with that infected chimpanzee. Once in humans, we just renamed it into HIV, and we had further mutations and further strain development, so now there are 3 different strains of HIV-1 in the human population, but all stemming from that kind of chimpanzee incubator, if you will, that allowed this virus to take hold. What makes HIV unique and so devastating is that HIV has a preferred host cell that it likes to infect. HIV's target cell for infection in humans is the helper T cell system of the human immune system. These helper T cells play a central role in our immune system, and more importantly, they are our primary defense against viruses. So this virus attacks and destroys the very cells we have in our immune system that help us fight viral infections. So HIV compromises the human immune system by destroying these human immune system cells. It's important to note that patients don't die from AIDS. AIDS is not a deadly disease. AIDS simply compromises the human immune system. What patients die from are secondary infections that they should have been able to fight off, that they would have been able to fight off normally, but they couldn't fight off now because of the severe depletion of their immune system due to an active AIDS infection. Things that wouldn't normally be a problem are problems now for that AIDS patient because of the damage done to their immune system. And finally, the flu, influenza. Somewhere between 5 and 20% of the U.S. population gets the flu every single year, and usually about 36,000 people die from the flu annually, typically young children and older senior citizens. The flu virus is capable of infecting birds and humans, also pigs or swine, and it is an RNA-based virus. But interestingly, it's not a retrovirus. What makes it not a retrovirus is that it does not engage in this reverse transcription step, and the flu virus does not integrate into the host genome. So the viral genome is RNA-based, but we don't reverse transcribe and we don't integrate, so it's not a retrovirus. There are three main types of flu viruses, categorized A, B, and C. A and B are the most common. A is further divided into two subclasses that are named based on the types of sugars that are displayed on the virus's surface. These sugars are abbreviated H sugars or N sugars. So based on which sugar is there, we give these viruses different names that you may in fact recognize, such as H1N1. When you name a virus H1N1, it's being named because of which sugars are present on which parts of the viral surface. That's what that name represents. But we hear these names in the media, and we associate them with these viral infections. Like HIV, the flu evolves very rapidly. For HIV, this rapid evolution is due to an exceedingly high mutation rate coupled with a very, very poor DNA repair rate when the viral genome is copied. For the flu, evolution is driven by two factors. One is the same. There's a high error rate whenever the viral genome is copied. But second, when a host is infected with more than one type of flu virus, and that's fairly common, different pieces of that RNA genome mix and mingle, creating brand new combinations of RNA genetic material in that co-infected individual. So we might have a human flu, a bird flu, and a swine flu. But if we have co- or tri-infection, we have a mixing and a mingling where we can create a viral particle that has some avian characteristics, some human flu characteristics, and some swine flu characteristics all in one virus. And in fact, this is what the H1N1 virus is, the swine flu virus, and it could be quite dangerous when you start mixing and matching viral pieces in this way. This new virus is going to have its own virulence and its own characteristics. Sure, 9 times out of 10, the virus is probably going to be worse rather than better, and we never hear about those viruses because they burn out. They die off because they're poor infectors. But when these combinations give rise to super virulent strains, which are extremely contagious and debilitating, we hear all about it because many, many people get sick with this brand new virus. As I've already said, pigs can be infected with their own flus. Those are typically referred to as swine flus. Pigs can also be infected by avian flus and human flus. So just as the chimpanzees were the incubators for the new and novel SIV viruses, pigs are oftentimes the incubators for these new flus, giving all of these assortments a place to occur. I hope you have some appreciation for the fact that trying to predict what the next flu will be next year is the same as trying to predict how certain pigs will be tri-infected with these 3 different viruses, and what parts of these 3 different viruses are likely to come together. That's a really, really difficult prediction to make. It's like trying to predict which 3 people are going to park in which 3 parking spots in the parking lot next year. You can try, but how close can you really hope to come? So we make flu shots for what scientists and clinicians think is going to be the next big flu of the next flu season. But quite often, we're wrong. And so the flu shot you get immunizes you against a flu that might have occurred, but didn't occur, and so you wind up getting the flu that year because the flu that is widespread and contagious is not the flu that they predicted for the flu shot. In any event, what we talked about today was the introduction of the idea of a virus simply as a genetic parasite. It really is not free-living. It has no real purpose or consciousness or sentientness other than replicating as much as it possibly can at the cost of the host cell machinery by hijacking that host cell machinery. Viruses exist solely to copy themselves. That's their only point and function. Viruses that affect bacteria are called bacteriophages, or phages for short. We describe the lytic cycle as the virulent bursting cycle of viruses and the lysogenic cycle as the dormant integrated cycle. We defined plaques, talked about what plaques were, cleared sections that phages make on lawns of bacteria due to bacterial cell death. We went back and redefined generalized transduction. We had described it in the previous lecture, but now we gave it that name, generalized transduction. This is when virtually any bacterial gene can be transferred by a virus that mistakenly loaded a fragment of the bacterial genome into its viral particle. We then talked about specialized transduction. We're very much like F' plasmids. The virus enters the lysogenic cycle, it integrates itself into the host genome, it stays there dormant for some time, it begins to excise itself out of the host genome, but when it does some, it takes with it some of the bordering bacterial DNA that was next to it at the point of insertion, and loads that DNA into the viral particle as well, allowing that DNA to be horizontally transferred. We talked quite a bit about how phage can be used for gene mapping, how we can spot and measure recombination between different phages. We talked about Benzer's experiment with the intragenic mapping. Looking at 2 mutations within a single gene, Benzer was able to spot and measure recombination between those 2 mutations, thus proving that the single gene was not the fundamental unit of DNA. This work, Benzer's work, provided the first molecular view of what had previously just been a genetic and inheritance field. And we ended with RNA viruses. RNA viruses, where we see reverse transcription and integration of a DNA-based RNA genome into the host cell, are referred to as retroviruses. Retrovirus examples are common cold, HIV. All retroviruses include the genes GAG, POL, and envelope. POL for reverse transcription and for making the integration more readily, and envelope for the viral proteins, the viral coat proteins. And we ended with some facts on HIV and the flu, just as FYI. It's always interesting to learn about things that we have a kind of a common sense layperson view on and put them into the perspective of the material that we're learning in class, I think. So, where this leaves us now is at the end of Unit 2. All of this material on viral genetics will and can be up for grabs for the next exam, Exam 2. That exam will be on October 30th, on Wednesday. So be ready for that exam. No other material will be introduced prior to the exam, so the end of this lecture marks the end of the Exam 2 material. We'll pick up again after the exam talking about gene mutations. What are different gene mutations? And very briefly, how can those mutations be repaired? But we'll save all that, and let's focus on preparing ourselves for the exam and learning all of the material that we have up to that point. As always, thanks for watching.